Thank you everyone for joining us and welcome to the Cancer Research Institute's webinar series, Cancer, Immunotherapy, and You. Today's date is Tuesday, September 13th, 2016, and the title for today's webinar is Multiple Myeloma Immunotherapy. Where are we? Before we begin, I'd like to thank our sponsors. This webinar series is made possible with very generous support from Genentech, as well as LabAnswer and its employees, with additional support provided by AbbVie, Celldex Therapeutics, NewLink Genetics, and Regeneron. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. CRI is the world's leading nonprofit organization dedicated exclusively to harnessing the power of the immune system to fight all forms of cancer. We fund scientists around the world at all stages of the discovery spectrum, whose work has led to significant breakthroughs in cancer immunotherapy treatment. Over the next 20 minutes or so, you'll hear from an immunotherapy expert about how these breakthroughs are changing the way multiple myeloma is treated. After the presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your questions at any time throughout the webinar by typing in the Q&A box on your screen. You can also tweet your questions to us using our handle @CancerResearch and the hashtag CRIWebinar. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website and our YouTube channel. Now it's my pleasure to welcome today's expert speaker. Dr. Alexander Lesokhin is an assistant attending physician on the Melanoma Service and Immunotherapeutics Corps and a member of the Ludwig Collaborative Laboratory at Memorial Sloan Kettering Cancer Center in New York City. This research focuses on the ability of cancers to evade the immune system and on developing novel treatments to reverse this process. Lesokhin leads a portfolio of clinical trials testing the clinical and immune effects of immunotherapies in blood cancers. He received his MD from the Albert Einstein College of Medicine and then went on to complete his residency and oncology fellowship training at Columbia University Medical Center and Memorial Sloan Kettering, respectively. Dr. Lesokhin, thank you so much for joining us today.
Thank you so much for having me. I'm very happy to talk about multiple myeloma today, and I'll start at the top just by introducing the disease. This is a disease of plasma cells that make antibodies. The cells are clonal in the disease, and we can detect the antibody as an extra protein shown here. This can exist as a precursor condition where we just find the protein but no end organ damage. It requires treatment when end organ damage affecting the bones, blood, and or kidneys becomes apparent. So this up top here represents the natural history of the disease. There's a precursor state that is observed, and then symptomatic myeloma that takes a relapsing, remitting course where there is initial frontline therapy that includes multiple drug options, generally a 3-drug regimen in 2016 for those able, stem cell transplant followed by maintenance in all patients, and then in either early relapse or later relapse, multiple treatment options as well. With the advent of some immune therapies more recently, particularly monoclonal antibodies that I will discuss, I think median life expectancy for this disease is getting better and better all the time. So I'm going to talk about immunomodulatory drugs. These are drugs that have been in our armamentarium for myeloma for some time, but they also have some immune modulating effects and act as, I think, foundational combinatorial agents that we can use with other immune-based therapies. There are other immune modulating drugs that I will not touch on too much, things like cyclophosphamide and melphalan, things that are routinely used in this disease and have immune modulating effects, but I will mostly not discuss. I will talk about monoclonal antibodies that were FDA approved the fall of 2015, these two antibodies that are really, I think, changing the landscape of this disease, and talk about things that are under active investigation and also very exciting in the future of myeloma. I won't talk about allogeneic transplant. This is a compatible donor's immune cells being transferred into those that have that are able to receive this therapy. There are limitations to this approach, although it is an immune therapy and can lead to durable responses in patients. There are limitations because of the age of the population. I also will not talk too much about vaccines. There are some very interesting vaccine studies ongoing that are listed here, but this is a complex area that, in the limitations that I have, I will not talk about. So just to start with immunomodulatory drugs, these have a long history in medicine that is not always good. Thalidomide was developed in the 1950s as a sedative for morning sickness, but quickly it was realized that there's significant teratogenic potential, and this was really one of the worst, I think, medical disasters that's on record, and was quickly shelved. The drug was empirically found to benefit patients with leprosy, and it was FDA approved for this indication in 1998. And then through research efforts in the laboratory in the 1990s, immune effects and anti-angiogenic effects were identified. And it was actually the latter effects, not the immune effects, that led to its first being tested out in multiple myeloma, where thalidomide turned out to be active and was ultimately FDA approved in 2006. This activity led to the development of derivative drugs with less side effects or more manageable side effects, and as I will point out in a minute, more immune effects. And these are lenalidomide and pomalidomide, and as I said, I think these are foundational drugs in myeloma that we will talk about further in a bit. So lenalidomide has anti-myeloma effects in multiple ways, directly on the plasma cells, on where the plasma cell lives, what we call the tumor microenvironment, but it also has effects on T cells. These are the immune cells that we think are important for controlling cancer outgrowth, as well as natural killer cells. So it can enhance T cell proliferation, enhance T cell cytokine production, enhance T cells' ability to kill tumor cells, and does similar things for natural killer cells as well. So why is lenalidomide a big deal? Well, it really has become a standard treatment program for frontline therapy, as well as for maintenance therapy. And here's just one study. This is in transplant non-eligible individuals. So this is elderly and frail individuals, a 2-drug regimen that compared lenalidomide with dexamethasone given in 2 different ways, continuously or for a defined period of time, relative to an older thalidomide-containing regimen. And what you can see here is that clearly the continuous regimen was beneficial, and not only in progression-free survival, but also in overall survival, where lenalidomide and dexamethasone was better than sort of older drug combinations with thalidomide. This really brought this drug into the fore in combination, and these days typically a 3-drug combination. So in addition to its use in frontline therapy, lenalidomide is very commonly used as maintenance because it's pretty well tolerated as an oral drug and can be given continuously. Here's one of the studies, but this is one of several, that really show very similar things, that those in a placebo-controlled fashion, individuals that received lenalidomide had a delay in their progression and improved survival outcomes. So this is really a standard approach. So these drugs are not all the same. Certainly, from direct anti-tumor effect, lenalidomide and pomalidomide seem to be more effective, but they also have more immunomodulatory properties. And so, if they have greater immunomodulatory properties affecting T cell and NK cell activity, then they're probably good. The idea is that they are good partners for immuno-oncology combinations. And this is borne out through several pieces of data that I will touch on. And first, I'm going to talk about this in the context of monoclonal antibodies. So these are monoclonal antibodies, are essentially proteins that our body makes antibodies, but in the laboratory we can also make antibodies that are targeted against specific molecules. And one that is really, I think, transformational is anti-CD38 antibody, and the one that's there's several that are actually in development. Daratumumab was recently FDA approved. Another one is SLAM-F7 targeting antibody, which was also FDA approved, but this one in combination with lenalidomide. So these antibodies improve responses, but really don't increase side effects in a significantly meaningful way. There are some infusion reactions with them, and there are some long infusion times that are issues. But I think beyond that, they don't overlap extensively in side effects with the other medicines that we use and are very effective, as I will show you momentarily. So as I said, elotuzumab was approved in combination with lenalidomide dexamethasone. It was based on this study where it was compared to lenalidomide and dexamethasone by itself. Elotuzumab by itself has very limited activity, but it does activate natural killer cells, and so lenalidomide, because it also does this in non-overlapping ways, was felt to be a good partner for this drug, and this turns out to be the case, as shown here. There was a delay in progression-free an improvement, in progression-free survival in those that receive elotuzumab. So this is now an approved drug for patients that have received 1 to 3 prior therapies and is actively being studied in the frontline setting. So what about the other monoclonal antibody, anti-CD38? This drug does have meaningful single-agent activity in people in this study that have had multiple prior therapies. This is really a very refractory patient population, and a proportion of these individuals respond in a durable way. And what's very interesting about this drug is that even those that don't seem to respond in terms of having a decrease in the amount of M spike that they have, that monoclonal protein I showed you in that very first slide, they still seem to do better than what would be expected for this heavily pretreated patient population. And there are some ideas around this, which I will touch on briefly in this slide. So we think that daratumumab has direct anti-myeloma effects because myeloma expresses this target CD38 at very high levels. It turns out that actually the immune microenvironment within which myeloma exists also expresses this target pretty significantly, and that CD38 targeting with this drug actually eliminates some of the immune inhibitory cells in that microenvironment. And these cells come in flavors: myeloid cells, suppressive T cells, suppressive B cells, and some subsets of these cells express the CD38 and are depleted, therefore improving the environment for an immune effect. And this is observed in that T cells, again, those are the immune cells that we think are good to control cancer outgrowth. Those T cells are expanded in a specific manner in those that seem to respond to daratumumab. And this, again, suggests that this would be a very interesting partner for another drug that modulates the immune microenvironment, such as lenalidomide, for example, or pomalidomide. That's another example. And this is demonstrated here in this phase 3 study that compared, and was recently actually reported out about 2 months ago, daratumumab with lenalidomide and dexamethasone relative to lenalidomide and dexamethasone alone. And what this study observed was, again, a really significant benefit in terms of progression-free survival in the daratumumab containing arm in combination with lenalidomide and dexamethasone. This was an early relapse setting, 1 to 3 prior therapies, and this actually supports the earlier use of daratumumab in our therapies. And frontline studies are also ongoing. So as you can see here, the plasma cell-directed monoclonal antibodies are really looking to transform the myeloma therapeutic landscape, and for the benefit of our patients, hopefully. So what about, I've talked about antibodies that target the myeloma cell. What about antibodies that target the immune system? What about drugs and antibodies that actually don't target the myeloma cell directly, but seek to modify how the host or how the patient responds, how the patient's immune system responds to myeloma? So the emerging field in this area is the field of immune checkpoints and agonists. And so these are a family of proteins that are displayed on an immune cell surface T cell. After the T cell sees its precise the precise molecule against which it responds. So T cells are very precise. It's a lock and key kind of interaction between the T cell receptor and the antigen it sees, and that turns the T cell on, and then subsequent signals through either checkpoints or agonists lead to activation or turning off of that T cell. And it turns out that this pathway is these pathways are dominant pathways in which cancers avoid immune detection. And the question that has come out is, the result of multiple clinical studies in a variety of solid tumor malignancies has suggested that these are extremely active drugs, and that therapeutically targeting these receptors is an extremely active clinical approach in cancer therapy. The question is, how does this affect, what is the role of these receptors and drugs in myeloma? And I will speak to that in just momentarily. So again, large family of molecules, inhibitory molecules that if you block them, you can lead to greater T cell activation once T cells have seen their specific target, or activating receptors that can be targeted with antibodies that further promote that activation. They're called agonist antibodies, and again lead to greater T cell stimulation. Not to belabor this too much, but I'm just going to show a specific example. So here's a T cell. It has a specific receptor called the T cell receptor. It sees its antigen on either a tumor cell, one of our own just regular cells, or on specific cells that are specialized to present antigen. They're called professional antigen presenting cells. Once it sees that, has that specific interaction, the T cells will secrete substances called cytokines that lead to an inflammatory process, inflammation, and then a counterbalance, a regulation, which leads to upregulation of the triggers for the inhibitory receptors activated T cells express. This, if you think about it, this system actually makes perfect sense. You have something that is attacking your body. Your immune system comes in to the rescue. It gets activated, and it causes inflammation. And in response to that inflammation, your normal cells will increase the expression of triggers that say, hey, I'm normal, don't touch me, don't hurt me, just get rid of the bad stuff and move on. And so this is a very potent mechanism of homeostasis that cancers use to avoid detection. We now know through a number of pioneering studies looking at patients with a variety of solid cancers like lung cancer and melanoma, where these drugs really work in a proportion of patients, what some of the framework is that determine who responds to these therapies. Patients that have T cells that see their tumor are susceptible. Where the tumors are actually presenting the antigen, those patients seem to be susceptible. And there is evidence of this homeostasis going on, where the tumor cells are being seen by the immune system and are protecting themselves through this PD-1 pathway. So these are the folks that respond. So because of this remarkable clinical activity, these drugs have been tested widely in many cancers, and there are drugs that target the anti-PD-1 molecule, the receptor that's on the T cells, and here's several of the drugs. And there are also drugs that target the triggers for that receptor, and that's called PD-L1, and these drugs actually block the PD-L1. So there's a couple of ways, and there's a lot of clinical studies, and actually the CRI has a cancer immunotherapy clinical trial portal that can help identify clinical trials for specific diseases that you can access to look for trials in myeloma with these various agents. I'm going to highlight just a couple of them here. So the first study that actually looked at PD-1 pathway blockade in myeloma was actually a signal-seeking study that looked at a whole host of diseases, including multiple myeloma, and 27 patients on the study were treated. This was with just anti-PD-1 by itself. Again, no drug to target the myeloma cell directly was involved here. And unfortunately, although there was some activity seen in some lymphomas and pretty striking activity in Hodgkin lymphoma, In myeloma, it was a little bit underwhelming. The best response was, or the most common response was stable disease, and it was relatively short-lived for many patients. There were a few outliers, but nevertheless, by itself, this doesn't seem something that we as physicians should be offering our patients. But as I've said a couple of times, IMiDs seem to enhance T cell and natural killer cell activity and antibody-dependent effects, and these are turning out to be really good partners for immuno-oncology combinations. So how do these combine with anti-PD-1? So there's been a couple of studies that have been reported at this point in national meetings, not yet really published yet, but here's one, and this is pembrolizumab. This is an anti-PD-1 drug as well, in combination with lenalidomide and low-dose dexamethasone. This was a study that accrued about 51 patients, and at the time that this was reported, 40 of them had been on treatment long enough to actually have a response assessment, and you can see that the majority of patients had some modicum of response. And what's particularly interesting about this is that this was a combination with lenalidomide, and there was a group of patients, a substantial group of patients, that were refractory to lenalidomide, and this is basically people that had previously gotten lenalidomide where it stopped working, or they'd gotten a lenalidomide-containing regimen and the disease unfortunately progressed 2 months after that. These patients still responded, a substantial proportion of them. So this clearly is some mechanism here where these two drugs synergize. And not just with lenalidomide, there's also another IMiD, pomalidomide. So pembrolizumab given with pomalidomide was also reported at ASH this year. And here again, in highly refractory patients, about 55% of patients responded. And it turns out some of these responses are actually quite durable. So I've talked to you about PD-1 blockade with IMiDs. What about with daratumumab? I also mentioned that there's direct on-tumor effect and there are immunomodulatory effects, and these seem to be non-overlapping with the way that the IMiD drugs work. And so this is also an area of active investigation, looking at PD-1 pathway blockade combined with daratumumab, as well as with immunomodulatory drugs, either lenalidomide or pomalidomide. And these are clinicaltrials.gov reference numbers listed here, but again, you can also find these in the Cancer Immunotherapy clinical trial link on the CRA website. So moving on to sort of, as opposed to looking at endogenous immune responses that patients may have already pre-existing, can we basically do the Frankenstein-like thing? Can we just take out their T cells, engineer them to respond to their myeloma, and treat them that way? And so this is a therapy that there's a lot of excitement about because there have been recent advances, and it's called CAR T cell therapy. And so how does this work? In general, patients undergo what's something called the pheresis procedure, where their immune cells are taken out and they're sent to the laboratory, where the cells, generally through a viral delivery-based mechanism, have a gene transferred into them that leads to the expression of this chimeric antigen receptor. And these are called CAR T cells. And these cells are typically reinfused into patients after some chemotherapy and for what's called lymphodepletion, this lymphodepleting conditioning. And I'll touch on this a little bit more in a moment. So the first sort of time that this was reported for myeloma, and I should say that these are therapies that seem quite promising in other diseases like ALL, and showed initial encouraging responses in CLL. But in myeloma, a CAR, or a chimeric antigen receptor targeting anti-B cell maturation antigen, was first reported this past December. And so let me just explain what a chimeric antigen receptor is. Basically, I mentioned that the T cell has a very specific receptor that makes it target just one thing. And when it sees its antigen, it sends a signal through this thing called CD3 zeta that's on the inside of the T cell. And then it requires additional signals, as I mentioned before, those checkpoints and agonists. And one of these sort of agonist molecules, the prototypical one, is CD28, and that sends that signal 2. So you have signal 1 and 2. And a CAR actually just takes, using DNA recombination technology that can be performed in the lab, it just takes the pieces of the T cell signaling machinery and the co-signaling machinery and puts it on the inside of a cell. And on the outside, it gets specificity by identifying an antibody that is specific for a particular target. So as I mentioned, antibodies are very specific for one particular thing. Our body makes these antibodies pretty often to prevent us from getting a virus or a bacterial infection. But we can also generate antibodies through vaccination. And so we can, in the lab, make an antibody against a specific target, then identify what that remove that area of specificity and just stick it on here onto this chimeric antigen receptor. So as I mentioned, this is a Frankensteinian kind of way of going about things, but it seems to be quite effective. And so this is what the National Cancer Institute generated CAR T looked like. They had the specificity from an antibody against BCMA, and then they had these various signaling molecules so here's the specificity, and these are the signaling molecules that were on the inside of the cell, and they took T cells out of patients and transferred this gene construct into them, made a CAR T cell specific for BMA, and gave it to patients at various doses in a dose escalation study. And what they saw was quite interesting, that patients had partial responses, some very good partial responses, and some complete remissions. So now this all sounds great, but when you have and it is great, I think, but when you have a very potent immune response directed against one thing, there's a potential for side effects. And one of the main things that we have observed in a variety of CAR T cell studies, which have been going on for several years now, is something called cytokine release syndrome. And this patients develop high fever and low blood pressure, and they get quite sick actually for a period of time and require close monitoring in the hospital for this syndrome. Now this is something that is expected, that we know how to manage, but it is a meaningful and relevant and very important side effect that requires specialized centers to deliver this therapy at this point. So this is an area of very active investigation at this point. So then finally, I just want to talk about really, these are actively being investigated now, but I think in the future we all understand that we need to be able to combine everything we have in order to be able to cure and totally eradicate myeloma. And so the question is can we combine traditional therapy, transplant that I mentioned, autologous stem cell transplant, the IMiD drugs, monoclonal antibodies, checkpoint antibodies, cell transfer with CAR T cells, vaccines? Can we combine it all to use the immune system to eradicate myeloma? And I mean, right now, today, we cannot, but there are ways to do this, and these studies need to be done in a stepwise fashion. And so this is one pioneering study that was done in 2005, where patients who were getting standard transplant, this is autologous stem cell transplant for myeloma, had T cells removed pre-transplant. So I should step back a little bit. Transplant is very good, it's very effective at treating myeloma and rendering patients into a minimal residual disease state. However, it does actually also eliminate a lot of the immune response, and T cells are absent for quite a long time. And so even though it can render patients to have a low burden of disease, where immunotherapy might work quite well, it sort of shoots itself in the foot in a way, because the immune system is also rendered to a low level. And so what these investigators did is they removed T cells through a standard apheresis procedure and then gave them back after transplant. And they used just a model of vaccine against pneumonia in this case, and demonstrated that if you didn't have the T cells infused after transplant, you couldn't get a response to a pneumonia vaccine. But if you had your T cells conditioned beforehand, they were removed and then reinfused early after transplant with additional vaccine, that patients could develop immune responses to pneumonia in this case. Now, we're not so interested in treating pneumonia, we're interested in treating myeloma. And so the idea here is, how do we use this kind of approach and as a platform for our other more active immune therapies now. And so this is a study that is due to open here and at another center in about a month or so, sponsored by the Ludwig, actually, and the MMRF, that seeks to sort of develop this type of framework. So I'm going to go back here. So the idea here is we're using transplant. We're going to condition patients with checkpoint blockade, take out their immune cells, and then after transplant, reinfuse those immune cells and give additional checkpoint blockade. And here there's a variety of schedules of checkpoint blockade. So here I'm going to say checkpoint blockade, I should be using this arrow, checkpoint blockade, lymphocyte removal, transplant, lymphocyte return with additional checkpoint blockade at various times, early or late after transplant, followed by PD-1 pathway blockade and IMiD maintenance therapy. And so the idea here is that we're building a platform that uses available ways that we have to, that we can then subsequently build on by conditioning these lymphocytes before returning them, either through CAR T cell therapy or some other intervention to be determined, or we can use, in addition to checkpoint blockade, perhaps we'll be able to use other monoclonal antibodies targeting myeloma. And so this is an initial step into that direction to evaluate, can we safely incorporate checkpoint blockade into this standard treatment paradigm. So I think touched on a lot about ways that we have available today to attack plasma cells using traditional chemotherapies. I haven't talked at all about proteasome inhibitors, but we have talked a bit about immunomodulatory drugs, monoclonal antibodies, and there's these other drugs to target the plasma cell. But I think as we can see, there are now a variety of tools available to us, immune checkpoints, CAR T cells, vaccines, allotransplant, to help the patient attack the plasma cell. And the idea is, can we combine these and really improve the future for myeloma in the coming years. And so with that, I think I'm open for questions.
All right. Well, thank you so much, Dr. Lesokhin, for that. It's good to see that there seems to be quite a bit of progress happening on the treatment front when it comes to multiple myeloma. That there's a lot of activity in terms of bringing together some of the latest treatments to come out of the immunotherapy research and bringing those to multiple myeloma patients, either alone or in combination. So thanks for walking us through that. You know, that last one of the last trials that you highlighted with the durvalumab and tremelimumab, I mean, I'm very fascinated by that trial. We're also one of the sponsoring organizations of that trial, so we're following that very closely. But the treatment plan is pretty complex, you know. And how do you explain that to a patient? And how does a patient come about, you know, trusting that this is you know, it is complex, but it's also cutting edge, highly promising science. How do you explain this to patients?
Yeah, so I mean, I think I have the benefit of that transplant. So if I were to just sort of go back to this slide, so high dose transplant is a routinely delivered therapy in myeloma, and so the processes of collecting stem cells are pretty something that patients are quite used to. And collecting lymphocytes is quite similar. It's the same process. So these are all processes that we routinely do. And then some sort of maintenance strategy is also something that we routinely do. And so I think the way I would explain this to a patient is that, you know, allogeneic transplant, where we take a donor transplant, where we take donor cells for that, donor stem cells, and we're inclusive of the immune cells that are present there and use them to render an immune effect to eliminate that minimal residual disease that's left over after transplant, is a therapy that may not be available for everybody. But if we can modify the endogenous response through checkpoint blockade or other therapies, CAR T cell therapies, other therapies, we can then essentially try to jazz up an individual's own lymphocytes to then reinfuse them early and perhaps get an autologous sort of graft, autologous response that's almost or somewhat similar to a donor-based transplant, that sort of immune effect here. So I agree, it is a complex study, but I think the idea is that we're just trying to restore an immune response in the setting where we think it would have the greatest chance to really tamp down myeloma.
All right. I would like to remind everyone who's watching that if you have any questions for Dr. Lesokhin, you can enter them into the Q&A box on the screen, and we'll try to get to as many questions as we can. Dr. Lesokhin, one of the attendees, a patient with what's called smoldering myeloma, is wondering how his or her patient population can benefit from immunotherapy protocols. First, what is smoldering myeloma, and kind of what's going on there? Is there an immunological component at play, and can immunotherapies help those patients?
Yeah, so smoldering myeloma is essentially a precursor to myeloma. That's where we can detect this clonal proliferation of plasma cells, where we can detect the protein in the blood, but there is no end organ damage or any myeloma-defining event in which the patient requires therapy. This is actually a very active space for investigation, because perhaps, if you're driving towards a cliff, and you're driving towards a cliff, then you want to stop before you get there. And this is also a space where individuals have not received a lot of the chemotherapies and immune suppressive medicines like dexamethasone that are such standardly used therapies in myeloma, and could potentially be sort of immunologically naive. And so there's a variety of vaccine studies that are being tested here. The monoclonal antibodies, I believe daratumumab is being tested here pretty extensively, actually. And there's also checkpoint blockade is also being tested in this space. So definitely this is an active area of investigation.
I was really excited to see some of the data that you showed with an 88% overall response rate in patients receiving the pembrolizumab and it looks like the lenalidomide and dexamethasone. So 88% sounds like awesome. Can you put that into some context? What does that really mean when a patient's trying to understand trial data and they see 88% response rate?
Yeah, so I think in myeloma, particularly at the time of relapse, minor responses are meaningful, but really what we're aiming for are complete eradication of disease. And in 2016, we can look at minimal residual disease and really look to see that if we can eliminate and get that minimal residual disease, even in the relapse setting. We know from frontline studies as well as from some relapse studies that really depth of response matters a lot. And it's not yet clear whether in the context of things like pembrolizumab, whether some modicum of response is enough, and you can render an individual into a state of equilibrium where their immune system seems to control the disease, or whether or not depth of response is really very important. So I think it's hard to say. These are early studies. That's very encouraging results, but we have to wait and see how sort of larger randomized trials pan out.
What's the estimated timeline on those trial results? When will they read out? Do you know?
So there are a couple of phase 3 studies that are accruing. I think in this setting it'll probably be 2 years, 3 years, around that time frame.
Okay, great. And you mentioned a few times, and I really appreciate this, that the Cancer Research Institute has a clinical trial finder service that's dedicated to immunotherapy clinical trials. And I'll be sharing some more information on that with the participants in just a moment. We have another question from Eric, who says, is there any evidence to favor CD19 over BCMA as a target for monoclonal antibody or CAR T cell therapy?
Yeah. So that's a somewhat controversial question. There is a CAR T cell study that got a lot of attention targeting CD19 for myeloma, and there was a patient that did respond. There is a hypothetical thought that although most plasma cells, myeloma cells, are actually do not express CD19, that maybe some of the sort of precursors or myeloma stem cells express CD19, and perhaps that even a little bit of CD19 expression is enough, because CAR T cells are actually extremely potent. I think that the BCMA target at this point has had more traditional responses and perhaps is a little bit clearly more likely to be developed than CD19 in myeloma. But I think, of course, that hypothesis of the, stemness is an interesting one, and I think we'll find out with additional study. I believe that center is actually exploring that further.
Great. So you touched on various checkpoint inhibitors, mostly the PD-1 and PD-L1, not so much the anti-CTLA-4. What can you say about anti-CTLA-4 therapy in multiple myeloma?
So there hasn't been very much study of anti-CTLA-4 blockade in myeloma to date. There have been a couple of early studies of CTLA-4 blockade after an allo transplant. Some myeloma patients were included in that. It's a very difficult patient population to evaluate. And there are studies that include CTLA-4 blockade as well as PD-1 blockade that have been accruing and will probably be reported soon.
Something else that was a theme when you discussing the kind of most exciting recent trials and the very promising trials that are now accruing or soon to accrue is that no cancer necessarily, or certainly multiple myeloma, is likely to not be taken down by a single agent. That combinations will play a very important role in getting those response rates up and increasing that durability of response. And also that it's not just the new therapies, that you can take existing therapies and modify how they're given to patients and potentially get better results. So I think that's really exciting. Is there anything else you'd like to say about that?
Well, I think, yeah, I agree with you. I think that is a very exciting area of research. I think the potential for combinations here is large, and the number of combinations is also large. So really the challenge will be is what is the bar to select one combination over another as something that you want to sort of move forward and really use as a standard approach in the broad patient community. And I think all of us, lose sleep over that question.
When there are, so many trials opening or options available, how does a patient navigate that and decide what is the best course of treatment for him or her? And what role do you play in helping them reach a decision?
So I think here, this is really a lot of it is a very specific discussion between the patient and their physician, and there's so many different factors involved, including a patient's health status, eligibility for any one or another clinical study, available standard of care options, logistics of delivering a clinical trial, which, as you point out here in this slide that's here, can be quite complex in some settings, and of course that's not for everybody. Some more frail individuals would not be eligible for this kind of study. They would need something that's a little bit easier to deliver. And so there's so many variables that it really needs to be an individualized decision taken together with, I would say, with your physician and hopefully someone who's at a center that specializes in myeloma.
Right. So it's a team of people that together with the patient and the patient's physicians ultimately makes that decision. Well, that's unfortunately all the time that we have today with you, Dr. Lesokhin. I hope we'll have you back in a couple years to talk about this exciting trial, and hopefully we'll see some more optimistic results coming from that. So just to wrap up, I'd like to thank everyone for participating in today's webinar. I'm just going to scroll quickly here through these. Would like to thank once again our very generous sponsors at Genentech, as well as LabAnswer and its employees, as well as thanks for the generous support from AbbVie, Celldex Therapeutics, Newlink Genetics, and Regeneron. So this is the Cancer Research Institute has a website called theanswertocancer.org, which is developed for patients and caregivers to explain the different types of immunotherapy and how they work for various types of cancer. A key feature of this website is the clinical trial finder that Dr. Lesokhin mentioned. You can call a phone number that you'll find on that website and be connected with a navigator who can help you find matches for trials for which you may be eligible, and then which you would, as Dr. Lesokhin explained, take to your oncologist and discuss to see if it's a good fit for you. There is no multiple myeloma information available on this website just yet, but you can get that information on the Cancer Research Institute's main website at cancerresearch.org, where we have a list under impacting all cancers. You'll see multiple myeloma, and we have a list of the latest advances, the latest scientific questions that are being asked, as well as clinical trials that are testing immunotherapies in multiple myeloma. Also, on Saturday, the 24th of September, Cancer Research Institute is hosting its first ever Immunotherapy Patient Summit here in New York City. Attendance now is full. I think there is a wait list. You can register for the wait list. But for those of you who either don't want to wait on the wait list or are unable to actually be in New York City, through generous support of a sponsor, we're able to broadcast the proceedings of the Patient Summit live on the internet free of charge. So you can go to bit.ly/immunotherapy patient summit to register for that and to get instructions for accessing the live stream broadcast. Again, that's September 24th, Saturday. We will also be video recording the proceedings, and those will be available on the Cancer Research Institute website. So I strongly encourage you to check that out. So with that, I'd like to say, we have another webinar coming up next month. Let's see, it's a breast cancer immunotherapy webinar. And I'm just looking for the date here. Yes, October 19th with Dr. Sylvia Formenti from the Weill Cornell Medicine and New York Presbyterian Hospital in New York City. You can register for that webinar as well as review this and other past webinars at cancerresearch.org/ you guessed it, webinars. And with that, I'd like to thank you all. Thank you again, Dr. Lesokhin, for your time, and I wish everyone well, and goodbye for now.